Today we will discuss about mechanical forces influence on immune response. Uh, Although we have covered some of this part in our previous videos, it's better to go through all this thing in a single attempt or in a single video. So we will start with this extracellular matrix force. So what happens when this extracellular matrix come and interacts with the integrin that are present on the immune cells is that the integrins will provide these mechanical forces to the effectins that are present on the immune cells, which is connected to the myosin 2, and it will act as a retrograde flow. That is, it will oppose the flow of the immune cells. If immune cells are flowing in these directions, then a retrograde flow will flow in opposite directions, and that will reduce the flow of the or speed of the immune cells. And next, in terms of this membrane tension, so these are the membranes which are not, these are the membrane of the immune cells which are not exerted, which does not exert any mechanical force because of the extracellular matrix. And when they exert the mechanical force on the extracellular matrix or under any other circumstances, that will lead to the opening of this pore, and it will allow the ion transport uh, in and out of the cells. So these things we have already covered. This second part we already covered in our previous videos. You can go through that videos to cover to understand how it will affect the intercellular changes within the cells. Next one is about the mechanical forces. So this is in this case, since the immune cells at the specific site or where the immune cells recruitment are required at the site of infections, the endothelial cells near the site of infections will release selectins, and that will attach to the immune cells and helps the immune cells towards these rolling adhesions or these ILP formations. And thereafter, because these immune cells will act as a invadopedia that we have discussed in one of our videos, where they will degrade the extracellular matrix and helps the helps the intra extravasations of the immune cells. Or this, in this case, here it is mentioned here as, here in this slide it is mentioned as transmigrations. So through this invadopedia they will be able to uh, transmigrate at the site where it is required through the expression of these selectins on near the endothelial cells of uh, near the endothelial cells, expression of the selectins on the endothelial cells near the site where the infection is being recognized by the host immune system. Next part is about these immune cells, how they interacts. The T cells interacts with these antigen presenting cells. However, in this case also the mechanical forces or these interactions can become more efficient when this LFA1 will interact with this ICAM1 or ICAM2 that are present on the dendritic cells, and this is the strong bond, and that will allow this T cell receptors and major histocompatibility complex interactions and exchange of informations, that is antigen informations from the dendritic cells about the harmful foreign bodies will be provided to the T cells through these T cell receptors and major histocompatibility complex interactions. And these interactions become uh, more prominent more prominent when this LFA1 and ICAM1 interactions occurs. So, and these are the strong interactions. So this will help the T cells to develop specific antibodies against the harmful foreign substances. Next one is about these B cell receptors. So in this B cell receptors, what will happen is the B cell receptor, the high affinity antibodies will be able to enter into these B cell receptors, and thereafter these B cell receptors will generate antibodies, or B cell receptors will generate the immune response. We generate the immune response against the harmful substance that has been detected by the immune that has been detected by the immune host system. Here also the interaction of the B cells and antigen presenting cells occurs. And in the case of this final panel F, the cytotoxicity T cell lymphocytes. So what it will does is it will form a strong synapses with the cancer cells, and thereafter it will release this perforin that will trigger the apoptosis in the cancer cells. This is also one of the kind of immune response that will leads to the apoptosis of the cancer cells, as perforin will trigger the apoptosis signaling pathways, apoptosis signaling pathways, and since they are these cytotoxicity T cells, T lymphocytes will release this perforin into the cancer cells. It is high likely that apoptosis will be triggered in those cancer cells. So I hope you got a just this brief overview about these mechanical forces influences on the immune system. And if you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.